Which? Okay, so our next speaker is Adria LeBoeuf, who comes from Israel, currently, present affiliation, and she will tell us about JH.
Hello. So right now I'm in Ofer Feinemann's group at the Weizmann, but the work I'm going to tell you about follows through on work I did in Laurent Keller and Richard Benton's group in Lausanne, and it's also a collaboration with Eyal Prifman's group in Haifa. Okay, so when is it evolutionarily advantageous for two individuals to exchange bodily fluids? So fluids tend to be exchanged only in the most fitness-relevant behaviors, things like mating or parental care, where an important component needs to be transferred from one individual to another. And often along with that primary component, like genetic material or food, there are a host of other components that are also transferred that can be a bit more manipulative or directive in nature. So seminal fluids are a great example of this. But the social fluid that I'm focusing on is trophallaxis fluid, so the fluid transferred during trophallaxis. So as all of you know, trophallaxis is a beautiful behavior that is exhibited in many, many social insects. And we often think about it as something that occurs between adults, but it also occurs between larvae. And actually in the definition of trophallaxis, that was the primary definition, was adult larva trophallaxis. And as I said, in many species, this is the primary way that all food can be transmitted throughout the colony. So we can think about trophallactic fluid as a kind of social circulatory system of the superorganism, in that it can connect each individual within the superorganism, all the way from the foragers to the larvae. Okay, great. Yes. So in late 2016, I published a paper on what this trophallactic fluid contains, focusing primarily on Camponotus floridanus. And we looked using many different techniques, and we found many different things. We found cuticular hydrocarbons, as had been predicted for years. We also found microRNA and other small RNAs as well, which we didn't tell you about then. And what was really surprising was that we found relatively abundant juvenile hormone. So juvenile hormone is a major insect developmental regulator. It controls many things, involved in many different pathways, but fundamentally it controls the larval molts, and then it is reduced to trigger metamorphosis. So this was a big surprise. We find approximately the same order of magnitude as we find in hemolymph. And then the study was primarily a proteomic study, and so we found many different proteins also in trophallactic fluid. We found many things involved in digestion, as expected. This is the crop. We also found many proteins involved in immune defense and many involved in growth and development, namely a family of juvenile hormone esterase-like proteins, which were highly abundant. So juvenile hormone esterases are the major enzyme that breaks down juvenile hormone to create these larval molts and eventually metamorphosis. So this was really exciting. And it made us think, okay, maybe we shouldn't be thinking about this adult-adult interaction, but rather something more like development. So we wanted to see if development could be manipulated by trying to manipulate the contents of trophallactic fluid. So we supplemented the trophallactic fluid with additional juvenile hormone by adding more juvenile hormone to the food of workers who were rearing larvae. And what we saw was a small effect on head size, but the more significant, more notable effect was more than double the number of larvae were reared through pupation and metamorphosis. So the same batch of larvae yielded many more new workers. So this led to what I call the crowdsourced development model, which is essentially that each individual within the colony perceives the colony's needs for growth or homeostasis, and they then feed this into their trophallactic fluid cocktail to adjust what they need in the next generation. This then leads to more or fewer larvae reared to adulthood, which can then feed back onto the social environment. So what I'm going to tell you about today is about these juvenile hormone esterase-like proteins and how they might be a mechanism that these ants are using to control their own development, okay? Or, well, the next generation. Okay, great. So just to give you guys a bit of context, juvenile hormone esterases have been very well studied, and canonically there's only one per species, and it's defined by the fact that it should be very specific for breaking down juvenile hormone, and it should have a characteristic expression pattern peaking in late larval development and in the adult fat body. Okay, so we wanted to find out, we wanted to look at the juvenile hormone esterase-like proteins we find in trophylactic fluid in a larger context. So we re-annotated all of the esterases in the Camponotus floridanus genome. I wish we'd had the new genome. And we've got 26 of them, and many of these are fragments. And they fall into 4 tandem arrays and then a variety of other little bits. One of the big tandem arrays down here we find is sparsely expressed in both hemolymph and larva. And only some of these have an intact catalytic triad. That means the residues needed to actually do the enzymatic activity. We found another small little array, which was more expressed in hemolymph. And I'm talking about protein presence here, and this is not expression, it's protein presence. And these two do not have the full catalytic triad necessary to actually do this enzymatic work. And then we have the trophylactic esterases, which are on 2 tandem arrays and 1 single gene scaffold. So, and all of these have an intact catalytic triad. So they look quite compelling and potentially involved in breaking down juvenile hormone. Okay, so we wanted to look in a bit of a broader context. Do we have the same pattern of JH and JH esterases in this fluid, in this social fluid, in other species? So we looked at the other 3 species where we had assessed the trophylactic fluid proteome, and we found that in Camponotus floridanus, Camponotus spela, another Camponotus, we also find an enormous amount of these proteins. So in both of these 2 species, it corresponds to 18% of the total endogenous protein we find in this fluid. It's a lot. While in Invicta and Apis mellifera, we find a few other esterases, but not particularly abundant and not related to these. We also wanted to find out, is there juvenile hormone in these other species? And we find the same pattern. So we find juvenile hormone in both of these 2, but not these. Okay, so we wanted to zoom in on what is this difference here. And the best way to do that is using something a bit more high throughput than these biochemical assays. And so we got a bunch of genomes and transcriptomes. And as many as we could find, but with an emphasis on formicine ants. We re-annotated all of the carboxyl esterases. And here are the species we're looking at. And so you can see that we specifically zoomed in on Camponotus with all of the transcriptomes we could get. And we've got 8 species of Formica. We threw these guys into a giant gene tree. And just to give you some frames of reference, this is the trophylactic esterases over here. This was that mostly larval and hemolymph tandem array. And this is the one that was more in hemolymph. And over here, this big clade in yellow is capped or crowned by the Apis mellifera juvenile hormone esterase. So it is the homologous enzyme that from Apis mellifera to this family of esterases. So focusing on this particular clade, we used maximum likelihood methods to look into positive selection. So this is just that yellow clade right here, headed by the Apis mellifera JHE. And the first thing that sticks out is, and this is myrmecine ants, formicine ants, Camponotus, and Formica. The first thing that sticks out is there's a ton of Camponotus esterases here. So there are actually 46 of them in just 6 species, while for the Formica we have only 10 esterases for 8 species. So we have seen a lot of duplications. Next, if you look for red branches, these are indicative of significant signatures of positive selection. So you can see, if you look just in the formicine section of the tree, you can see that there's nothing over here, and many branches, half of the branches we detected are in the Camponotus section of the tree. Now, to add on the protein presence information that we have, here everything that's marked in bold are the species where we've looked in trophylactic fluid proteomically. And what you see is that as we get more derived, longer with increased branch lengths, and more positive selection, we have more abundant esterases. So these are the top 2 most abundant esterases in each of these 2 species. So these 3 things all go in the same direction. And now after positive selection analyses, we can look into what are the specific amino acids that have actually changed in these proteins. So looking at these 4 branches that make the difference from the more ancestral esterases to the more derived. And luckily, we have a structure. So juvenile hormone esterases, the juvenile hormone esterase from Manduca sexta has been crystallized. Inside its binding pocket, we have OTFP, which is a transition state analog of juvenile hormone. So it binds covalently. And here the catalytic triad are in yellow. And here, if we overlay the positively selected residues onto this protein structure for the Camponotus version, we can see that they are found around the exterior of the protein and also sort of behind the catalytic triad. And then one more little juicy bit is this one, which in the ancestral proteins we have a glycine. Here in the ant protein we have, and this is only in the most abundant trophylactic esterases, we have a phenylalanine. It's bulky phenylalanine at near the opening of the binding pocket. So altogether, these types of changes are somewhat consistent with the change in conditions that these things have undergone. As they've moved from the hemolymph, which is pH 8, to the trophylactic fluid, which is at pH 2 or 4. It's very low pH in formicine ants. So this corresponds to a large change in protein conditions. Now, many of these things correspond to this idea of neofunctionalization. So we have duplication, positive selection, a change in localization. Can we see a change in activity? So to assess this, we use that transition state analog, that OTFP, because each of those trophylactic esterases, if you remember, all had an intact catalytic triad. So this should be able to manipulate all of them at once. So we use the same sort of structure that we use to look at the effect of JH on development. And here we looked at JH, OTFP, and then both together. And then over time, we collected the pupae and checked them out. So this is what we saw. You can see that both JH and OTFP had effects, increasing the number of larvae that were reared to pupation, and they had a cumulative effect. So both together gave us even more larvae reared to pupation. On the other hand, we did not see an effect of OTFP on the size, only on their likelihood to undergo metamorphosis. Okay, so in essence, we can see that this juvenile hormone esterase inhibitor does indeed influence development. So to summarize, for these esterases, they have an intact catalytic triad. They're orthologous to the only known Hymenoptera juvenile hormone esterase. They've undergone duplication and positive selection. And finally, we see that larval development can be influenced by an inhibitor of juvenile hormone esterases. So all in all, I think there's still plenty of work to be done to explore the system. But I think this is moving into a direction where we can understand better how development can be crowdsourced by the colony. Thank you very much to everyone involved.
Thank you, Adria. I think we have time for a couple of questions.
So the effect of a juvenile hormone on metamorphosis is specific in the stage that the larva is occurring. So it has to come and to prevent metamorphosis. Do you think that they can actually regulate the time that they secrete the juvenile hormone and juvenile hormone esterase, or that it's always the same amount?
That's a great question. And actually, the work that I'm doing right now in Ofer Feinemann's group is more on when feeding occurs and how that's related to growth. And so hopefully that'll give us some answers to that, because I think it really is about feeding, the timing of feeding, because they don't feed very often.
Hi. Could you maybe speculate about why there's been such a huge expansion of these genes? What are they all for?
Yeah, absolutely. I think they've had to do a lot of changing. So there are a lot of different ideas that we could explore here, but there's been a lot of change over the different esterases, I mean, with all of these branches of positive selection, and it seems that all of the Camponotus species we have many, which suggests that maybe these duplications were somewhat ancestral. Many of the esterases are much less expressed than the few that are extremely highly expressed. Another possibility is that they could be different knobs and dials to influence development. Maybe they're differentially, one of them is expressed in eggs. Maybe these are trophic eggs, it depends on the sample, but yeah. So there are lots of different possibilities here, and I'm excited to find out more.
Thank you, Adria. I think we need to go to the next speaker.